Hello everyone, welcome to our third lecture in our video series on physiology titled Membrane Physiology, the Nerve. In this particular lecture, we will look into how transport of various substances occur across the cell membrane and see how this cellular transport, the charge of the cell membrane, as well as the concentration gradients of different substances across the nerve cell membrane together play a role in nerve signal conduction across the body. To start off, we'll revisit our concept of the cell membrane. As we have seen in the last video itself, the cell membrane is a lipid bilayer with various proteins and glucose molecules attached to the cell surface. The sugar molecules form the glycocalyx on the outer cell surface, and the various integral proteins and the peripheral proteins help in transport as well as other enzymatic activities of the cell membrane. But as we have seen in the last video itself, since since the cell membrane has a lipid bilayer nature, water soluble substances cannot easily diffuse through the membrane itself. Further, lipid soluble molecules can easily diffuse through the lipid membrane and pass into the cell. So this particular video will start off with discussing various forms of membrane transport that occurs across the cell and how various substances are able to get into and are able to exit the cell as well. So the type of membrane transports vary by a wide margin. They can include a simple diffusion, where diffusion occurs right through the lipid membrane, as in case of lipid soluble molecules, or simple diffusion through water-filled channel proteins, where water-soluble molecules pass through. There is also facilitated diffusion, about which we will learn later on. And finally, there is active transport. The diffusion part of the membrane transport occurs along the concentration gradient of the substance, meaning if the concentration of the substance outside is low and inside is high, then the particular substance will try to diffuse out of the cell. But instead, active transport occurs against the concentration gradient, meaning if the concentration of the substance is high on the outside and low on the inside, the active transport will try to pump more of the molecules outside of the cell to increase the concentration gradient, and thus energy is required for the whole process. To start off, let us look at diffusion through the cell membrane. Diffusion through the cell membrane is divided into two parts: simple diffusion and facilitated diffusion. Simple diffusion, as we know, is the kinetic movement of molecules across the cell membrane due to concentration difference. That is, movement of molecules along the concentration gradient. This particular phenomenon occurs due to kinetic movement of molecules, because if we consider a group of molecules towards the left side of the screen here, which are very concentrated compared to low concentration of molecules on the right side of the screen, then the kinetic energy of this molecule, the molecules on the left side of the screen will try to push them towards the right side. And thus we have mass movement of molecules from the left side to the right. And this is what we mean by movement along the concentration gradient. As you can see in this picture itself, this is an example of kinetic diffusion of a molecule. And this process, as we can see, is completely random. Next, we look into facilitated diffusion. Facilitated diffusion occurs when particular proteins help a particular molecule to move along the concentration gradient. A very good example will be in the case of water-soluble molecules, which cannot move directly to the lipid membrane. Such molecules need special proteins known as carrier proteins to bind to them and shuttle them across the membrane. Even though the concentration gradient allows them to move, the lipid bilayer prevents them movement. And as such, the carrier proteins act essentially like a bus on which the molecules can attach to and then move into the cell. One thing to note is that the diffusion process requires no energy or very less energy from the cell in order for the membranes to be transported across the membrane, for the molecules to be transported across the membrane. Next, let us look deeper into simple diffusion. Simple diffusion can occur simple diffusion can be divided into two halves. One for lipid soluble substances diffusing directly through the lipid bilayer, And another part is the water and other lipid insoluble molecules that pass through protein channels. Lipid soluble substances such as oxygen, nitrogen, and alcohols can dissolve right through the lipid bilayer and diffuse into the cell. Thus, they need no external help in order to move into or out of the cell. Water soluble molecules, on the other hand, cannot pass through the lipid bilayer, and thus they require protein pore channels. Protein pore channels are essentially cylindrical proteins that cross the whole cell membrane. These are integral proteins and they have a water-filled lumen. We can consider them as cylinders having a water-filled interior. Since there is water inside the particular pore channel, water-soluble molecules can easily pass through this channel into the cell. But these protein pore channels have two specific characteristics that help them prevent unnecessary entry of foreign molecules into the cell. These include selective permeability to certain substances. That is, the protein pore channels can allow only specific molecules to pass through them. For example, the aquaporin protein pore channel only allows water molecules to pass through it. And secondly, protein pore channels can be opened and closed by use of gates. We will learn more about what these gates are, but essentially the cell can choose to open or close the channel and allow or prevent the entry of various molecules through the protein pore channel. So let us first look at the selectivity of these channels. So to start off, we have to understand that the extracellular environment as well as the intracellular environment is hydrated. That is, there is water everywhere. So if we consider the example of ions, say potassium and sodium, they will not exist as just the ions themselves, but they will have a hydration shell of water molecules about them. Sodium and potassium have a crystal radius of 0.095 nanometer and 0.133 nanometer respectively. Since sodium, which is shown in the bottom of the screen right here, is much smaller but has the same charge as potassium, the localized charge on sodium is way higher than potassium. Localized charge is the parameter that is considered. Localized charge is the parameter that guides the size of the hydration shell of a particular molecule. Since the localized charge of sodium ion is way higher, therefore the hydration shell of sodium is much larger than that of potassium. Thus, in effect, sodium in a hydrated condition acts or behaves as if it is much larger than potassium itself. So the question is that how does the cell use this particular characteristic of ions in order to distinguish them and allow the specific ions to pass through protein pore channels and prevent others? To look into the same, we will take the example of sodium protein channels. The sodium channel is only 0.3 nanometer into 0.5 nanometer big and has a negatively charged inner surface, which is shown right here. When the gate of the channel is open, the hydrated sodium ion can enter into the channel itself. But since there is a high negative charge in the interior of the channel, water molecules are pulled away from the sodium ion, and thus only the naked sodium ion passes through the channel. The inner construction of the gate itself, of the protein pore channel itself, is made so that only the naked sodium ion can pass through without its hydration shell, and thus sodium channels are very selective for sodium ions themselves. Let us take another example, potassium channels. Potassium channels are 0.3 nanometer into 0.3 nanometer big. That is, they are smaller than sodium channels, but they do not have any charge on the surface. When a hydrated sodium and a hydrated potassium ion approach the channel, the sodium ion now is too large to pass through the channel, because as we have seen, the hydration shell of sodium is way larger than potassium. Thus, potassium in the hydrated condition acts as a smaller molecule and can easily pass through the pore channel, but sodium ions are blocked. Thus, potassium channels are selective for potassium ions. This is how the selectivity of various protein channels are maintained. Next, let us look into the gating of protein channels. The gating or the restriction of entry of various substances through the protein channel can be divided into two halves. One of which is voltage gating, the other is chemical gating. Voltage gating is the opening or closing of the membrane channel depending on the electric potential of the cell membrane. As we know that the cell membrane itself is inherently negatively charged due to the glycocalyx layer on top of the cell membrane. But depending on the amount of charge on the cell membrane, voltage gated channels can open or close. When potential of the cell membrane changes, the molecular conformation of the gate or the molecular bonds in the protein channel itself change, which allow closing or opening of the voltage gated protein channel. On the other hand, chemical gating is the opening or closing of membrane channel depending on the binding of a ligand. This ligand can be a hormone or other moiety that binds to the exterior of the protein channel and allows the channel to open. The binding of the ligand to protein leads to conformational change or chemical bond change in the channel, and thus it can open or close in presence of the particular ligand. There is another third type of gating that is present, which is known as mechanical gating, and that depends on the pressure exerted on the cell membrane. If a particular cell membrane is pushed or pulled in a particular direction, the protein gated channel can then open in response to that stimulus. That is the third type of gating that we are not going to discuss in this particular part. So this is the example of the voltage gated channels. As we can see, when the exterior of the membrane is positively charged, the channel remains closed. But as there is a change in the membrane potential, that is the exterior becomes negatively charged, the protein gated channel is now open. Similarly, for chemical gating, when there is no ligand binding to the gated channel, the channel is closed. But once the ligand binds to it, the channel is now open and allows the passage of various molecules through it. Next, let us look into facilitated diffusion. So we have already discussed simple diffusion, and now we'll move on to facilitated diffusion. Facilitated diffusion, as we have discussed, requires a carrier protein which helps in the transfer of the molecule from outside of the cell to the inside of the cell. Carrier mediated diffusion via specific carrier protein to help diffuse through the cell membrane is known as facilitated diffusion, which is shown schematically right here. So the transported molecule first binds to part of the carrier protein, and after its binding, conformational change occur in the carrier protein itself, which allow the transported molecule to enter into the cell. The rate of the diffusion here depends on the amount of the carrier protein present, and it also increases with the amount of the diffusible transported molecule present. So what does this mean? In case of simple diffusion, we know that if the amount of diffusible substance increases, then the rate of diffusion will also increase. It is also the case in facilitated diffusion, meaning that if the transported molecules amount increases, then a higher rate of diffusion we see. But this rate has a particular limit. The rate depends on the rate at which the carrier molecule can change between states. What does that mean? So we have a carrier protein which undergoes two states. One is the binding, and second is the release, which allows the transported molecule to enter into the cell. So this rate at which the carrier protein can can change from the binding to the release state and back to the binding state is what determines the rate of diffusion. So if majority of the carrier proteins are in the release state, they cannot bind to new molecules and allow their passage. And there is where the limit of the rate of diffusion occurs. But in case the carrier protein changes between states very fast, like it can bind and instantly release the particular transported molecule into the cell and then again change to the binding state, then the rate of diffusion will increase manifold. This is shown here in this particular graph, where we can see the rate of simple diffusion increases with increase in concentration of the substance. So greater the amount of substance, greater will be the diffusion. But in case of facilitated diffusion, we have a Vmax, or a maximum limit to the rate of diffusion, which depends on this particular phenomenon, wherein the carrier protein changes between states. So if the carrier protein changes its state very fast, then we will have a higher Vmax. Whereas if the carrier protein is slow between changing back from the release state to the binding state, then will have a low Vmax. Next, we will look into various factors which affect the diffusion rate. These factors include concentration gradient of the particular substance across the membrane, which is given as the difference between the outside concentration and the inside concentration, CO minus Ci. Next, it also depends on the membrane electrical potential or Nernst potential of the particular membrane of the particular substance. So what does this mean? So we know that the cell membrane by itself is inherently charged, but also the ions of the molecules which want to enter or exit the cell are also charged themselves. This charge interaction between the cell membrane as well as the intracellular environment as well as the extracellular environment is what helps. Let us start again. So membrane electrical potential or Nernst potential is what is another factor which affects the diffusion rate across the cell membrane. As we know, the cell membrane itself is inherently charged, and there is charge present in the intracellular environment as well as extracellular environment. The particular molecules that want to diffuse into or out of the cell are also charged. So this charge interaction, or the electrostatic interaction that plays a role in diffusion, is given by the Nernst potential. Nernst potential is described as EMF which is measured in millivolts and is given by the formula plus minus 61 into log of C1 by C2, or the concentration difference of the particular molecule outside and inside the cell. We will look into how this plays a major role in nerve signal conduction across the cell. Finally, we have pressure difference across the membrane, which also plays a role in rate of diffusion. This is given by PO minus PI. That is, if pressure is exerted on the outside of the cell, then lot of molecules will start diffusing into the cell because there is a directed motion or a force pushing them toward the cell membrane. Next, let us look at these concentration gradients that we were talking about. Here we are given the concentration gradients as concentration of a substance in the extracellular fluid and concentration of the substance in the intracellular fluid. As you can see, some molecules such as sodium and calcium are abundant in the extracellular fluid and very low in the intracellular fluid, with sodium having a concentration of 142 milliequivalents per liter in the extracellular fluid, whereas there is a very low amount, about 10 milliequivalents per liter in the intracellular fluid. But on the other hand, molecules such as potassium has very low concentration in the extracellular fluid and is present in huge concentration inside the cell or in the intracellular fluid. We will see how this also plays a major role in nerve signal conduction and maintenance of how these concentration gradients are maintained in the cell. So next we move on to active transport. Active transport is the last of the membrane transport phenomenon that we are going to look into. Active transport is the transport of a particular substance or molecule against the concentration gradient. Meaning, as we can see right here, active transport is essentially the transfer or transport of various molecules like potassium, which is present in higher concentration in the extracellular fluid, against its concentration gradient into the cell. Since this particular process acts against the concentration gradient, it requires energy in the form of ATP. Active transport can be divided into two halves: primary active transport and secondary active transport. For example of primary active transport, we will look into the sodium potassium pump. The sodium potassium pump uh, utilizes ATP in order to transport three molecules of sodium ion out of the cell and allow the entry of two potassium ions into the cell. Thus, we have movement of sodium, which is present in high concentration inside the cell, to the outside, and the reverse in the case of potassium. Both of them moving against the concentration gradients. This whole process requires ATP, and thus the sodium potassium pump has an ATPase activity as well, which utilizes the energy released in order to transport these ions across their concentration gradients. The sodium potassium pump is also an electrogenic pump, which means that the sodium potassium pump inherently introduces a charge gradient across the cell membrane. As we can see, since three sodium ions are pushed out of the cell while two potassium ions are allowed to enter into the cell, we actually observe removal of a positive charge from the interior of the cell. And because of this movement of a positive charge from inside to the outside, we have a negative charge accumulation inside the intracellular environment. Thus, the sodium potassium pump is also known as an electrogen. This was a primary active transport in which the molecules themselves are moving against their concentration gradient. There is a secondary active transport as well, in which one molecule moves along its concentration gradient and a second molecule just piggybacks on top of the first molecule and moves against its concentration gradient. For example, we will look into the sodium glucose pump. For the sodium glucose pump, the sodium ion moves along its concentration gradient. Sodium ion concentration on the outside of the cell is way higher than the inside of the cell, and thus the sodium ion wants to move into the cell. The opposite is true in the case of glucose. Glucose is present in minimum amounts in the outside on the outside of the cell compared to the inside. The movement of sodium occurs inside of the cell by binding to this co-transporter, which is a protein. But the movement cannot occur till a glucose molecule also binds to the co-transporter. Once a sodium binds to the sodium binding site and the glucose binds to the glucose binding site on the co-transporter protein, then only are both of them allowed to simultaneously move into the cell. Thus, the energy released to move the sodium ions into the cell is utilized in order to move the glucose molecules against the concentration gradient into the cell as well. This is known as co-transport. There is another form of transport known as counter-transport, and both of them are co-transport is also known as symport, and counter-transport is known as antiport. Like the proteins, the co-transport proteins are known as symporters, and counter-transport proteins are known as antiport.